So building off of our last lecture, we're going to talk about the primary signals in the transduction pathway in skeletal muscles. And in this case, we have 4 primary signals. And keep in mind that these signals are coming from the stimulus of exercise, and that the training adaptations that are going to occur in the muscle fiber are the result of an increase in the amount of specific proteins that are going to be produced in response to this training stimulus. Specifically, transcriptional activators are responsible for turning on the specific genes that are going to synthesize these new proteins in response to our exercise stimulus. The amount of these new proteins is going to be dependent on the intensity and the duration of the activity, as well as whether we're participating in resistance or endurance training, because they are going to cause different adaptations in the muscle. The first of our primary signals is mechanical stretch. And this primary signal is very sensitive to both the intensity as well as the duration. So it's sensitive enough to know whether it's low contraction strength over a prolonged period of time versus very high contraction strength over a short period of time, like you would have with resistance training. And specifically, when you have those really high loads of mechanical stretch of the muscle on the muscle membrane during resistance training, that's going to cause that primary signal to then send a cascade of signaling to promote contractile protein synthesis, which is going to lead to muscle hypertrophy. Calcium plays an important role in muscle contraction. We know it's released from the sarcoplasmic reticulum, so it can bind to troponin to allow for cross-bridge cycling. Importantly, the amount of calcium is different between endurance and resistance exercise. During resistance exercise, the muscle will have high amounts of calcium for prolonged periods of time, and it will have high amounts of calcium for short periods of time during resistance training. And that calcium is going to stimulate various enzymes within the body. One enzyme specifically is calmodulin-dependent kinase. And this enzyme plays an important role for a lot of the downstream effects that are going to occur through both endurance and resistance training. Additionally, during exercise, we produce free radicals. And these free radicals are actually an important response to training, which is why we don't want to blunt some of that inflammatory response that we have through training. It is part of a signaling cascade that leads to those downstream physiological changes, things like increase in mitochondria or hypertrophy within a muscle. Our last primary signal has to do with the energy balance within the muscle. And when the muscle is burning through its energy, it's going to stimulate AMPK. And if you haven't heard of this kinase before, it's adenosine monophosphate activated protein kinase. And this kinase in particular plays a really important role in how the muscle cell has homeostasis of its energy, and specifically activating glucose and fatty acid uptake, and then the breakdown of those molecules through oxidation, so when oxygen is present, when cellular energy is low. That brings us to our secondary messengers in the skeletal muscle. Remember, this is kind of like a game of telephone in the muscle as one enzyme communicates with the next. So we've already mentioned AMPK already, and its role that it's going to have in metabolism, specifically utilizing glucose and fatty acids, but it also contributes to mitochondrial biogenesis, which just means the production of more mitochondria within the muscle in order to be able to undergo more oxidative phosphorylation. PGC1 alpha stands for peroxisome periphylator activated receptor gamma coactivator 1 alpha, which is certainly a mouthful. So we can stick to the acronym in this case because that's how it's commonly referred. Importantly, this is the key regulator of mitochondrial biogenesis, and it plays a really important role in all things related to increasing mitochondria and then oxidative metabolism of the muscle. So it'll increase the number of mitochondria. It will support the shift in fiber type from type 2 fibers to type 1 fibers that are more oxidative, or type 2X fibers to type 2A fibers that are also more oxidative in this case. It's also going to promote angiogenesis, so an increase in the number of capillaries around the muscle to facilitate pulling oxygen off at the level of the muscle. PGC1 alpha is activated by 2 different enzymes. It's activated by P38, which is a mitogen activated kinase, as well as by CAMK, which stands for calcium calmodulin dependent kinase. And these enzymes specifically are really important for increasing the levels of calcium within the muscle. And they're responding to that in order to signal downstream to the PGC1 alpha, which has its effect on the oxidant properties of that muscle tissue. Calcineurin is a phosphatase enzyme. So it removes phosphate groups from molecules, and it's activated anytime you get a robust increase in calcium within a muscle. And it plays an important role in fiber hypertrophy. Again, that transition from fast fiber types to slower fiber types that behave more oxidative in nature and can utilize oxygen for metabolism. NF-kappa beta, or another mouthful which stands for nuclear factor kappa light chain enhancer of activated B cells, is another really important downstream molecule within our cells, and specifically in respect to muscle adaptations, it promotes the formation of antioxidants within our muscle to handle all the oxidants that we are now producing, specifically those free radicals. mTOR is our final second messenger, which stands for mammalian target of rapamycin, and it's a protein kinase that regulates protein synthesis and cell growth within the muscle. So this is the key regulator of muscle hypertrophy in response to exercise training. This slide is an excellent summary slide, and it shows you all the important components of those primary and secondary signals. So notice which primary signals signal to the secondary signals. And importantly, what is that keystone modulator? That's going to be our PGC1 alpha molecule. Importantly, this transcriptional factor plays a key role in many of the adaptations that we see with the mitochondria, or mitochondrial biogenesis. It's important to know the time course of these signals. You'll notice that some of the primary signals only take seconds, whereas some of those secondary signals may take minutes. Once we get to that key regulator molecule, the PGC1 alpha, that's going to take hours until we see an increase in activity of that molecule that's going to cause those downstream protein changes that are going to affect our mitochondrial biogenesis. Now keep in mind, just like when we're overloading the system so that we have these physiological adaptations, our body over time will adapt to that stimulus. And the same thing happens here. Our body actually adapts to the stimulus as we become more trained. So that means our sympathetic nervous system is going to release less epinephrine and norepinephrine, and we're also going to have blunted cardiorespiratory responses. Our heart rate will remain lower and our ventilation will remain lower. This is like that example of when you're out running with a friend and you're really annoyed because you're out of breath and can't talk, and that friend just seems to be chatting away because they're adapted and highly trained potentially then to that exercise, so they're having less of a cardiorespiratory response. Their body is better able to take up oxygen due to the adaptations of training. And so over time then, you're going to have less feedback from the muscle because the body is adapting and better able to utilize that oxygen. It also means that you don't have to recruit as many motor units to do any given activity. So we also become more efficient in that regard. And we know this because whenever we've trained individuals just with one leg or one arm, we don't see those same adaptations transferred than when we assess the other arm. So importantly, it is not whole body adaptations. It is specific to the regions of the body that are trained. So if you only train your upper body, you only get adaptation in your upper body. If you only train your lower body, you only get adaptation in your lower body. So this is one of those important reminder, folks. Don't forget to go to the gym on leg day. Keep in mind, there's both peripheral and central control of our cardiorespiratory responses to exercise. From our peripheral feedback, we specifically get information from group 3 and group 4 neurons that are within our muscles. And they tell us important things about how hard our muscles are working so that it can modulate both our heart rate, how hard our heart is working, as well as our respiratory centers. These neurons will respond to things like temperature, tension, and chemical changes within the muscle. Importantly, all of these sensory signals, these afferent signals, are going to come to the central nervous system to be processed for an efferent signal to be sent back out. We also have central centers that help with our modulation of our cardiorespiratory centers. Areas like our motor cortex, our basal ganglia, our cerebellum, all receive information and send out signals about how to modulate our cardiorespiratory centers based on the intensity of exercise. This is going to be very important for modulating both our muscle recruitment as well as all of those cardiorespiratory centers and how we're going to change the functioning of the heart as well as the respiratory system. These next two figures highlight the concept on the previous slide. The one on the left shows the peripheral control mechanisms in muscle and how they influence heart rate, ventilation, and kidney and liver blood flow in response to submaximal work and how those are affected and changed. The figure on the right shows the central control of motor unit recruitment, heart rate, ventilation, and liver and blood flow responses to submaximal work. So it shows what stimuli or inputs are affecting those outputs. One of our training principles is that of reversibility. So if you don't continue your training, or you stop your training, or you back off the intensity far too much, this will lead to detraining and someone's VO2 max will go down following any sort of adaptations that they may have seen from a robust training stimulus. One of the first things that you'll observe is a very steep decrease in the maximal stroke volume, so that amount of blood that can be ejected from the heart with each beat. And this is because of a loss of plasma volume. That plasma volume adaptation is a very transient adaptation to training, so you'll lose it very quickly if you stop training. Additionally, your tissues won't be able to pull off as much oxygen and utilize that for aerobic respiration. This occurs because the number and the size of the mitochondria goes down, which then lessens the oxidative capacity of the muscles. And you'll actually see a shift back from the slow fiber type towards the fast fiber type again that aren't as oxidative and have to pull from your glycogen stores to even be functional. Now keep in mind, reverse of the way we learned it as a training adaptation, the initial decrease is going to be from the loss of stroke volume, and then that later decrease this time is going to be from a decrease in the AVO2 difference. And when we learned that as a training adaptation, initially we had an increase in stroke volume and then later the increase in AVO2 difference. So it really shows you which one will adapt very quickly and which one adapts slowly. And the same is true as you're losing those adaptations. If you've ever done some sort of endurance training, you know it takes a lot of hard work to see these adaptations and improvements in your cardiorespiratory fitness that are largely because of the changes in the mitochondria. However, keep in mind that you can lose these adaptations very quickly through detraining. Within just 1 week, you can lose 50% of the adaptation that you had gained through training. Another 2 weeks, and now you've lost almost all of that adaptation. And what's even more difficult is it can take 3 to 4 weeks to regain that improvement. So we want to make sure whenever we're prescribing exercise and people are undergoing training programs that they do maintenance. When you work really hard to gain these adaptations, that you do some level of work all the time to maintain those adaptations so that we don't have detraining occur.